Yes, welcome to the second talk in our new virtual modeling biocomplexity seminar on cancer invasion and progression. My name is Andreas Deutsch. Together with Lutz Brüsch, I'm heading a research group on mathematical biology with a focus on medical applications at the Center for Information Services and High Performance Computing located at Technische University Dresden, Dresden University of Technology. The virtual modeling biocomplexity seminar team is completed by Robert Müller and Simon Züger, who are both also members of our research group. I would like to thank the head of the Center for Information Services and High Performance Computing, Professor Wolfgang E. Nagel, for generous support of our seminar idea. We have registered participants from more than 20 countries. What you see here on the right-hand side is not the new corona cases, not a WHO graph, it's the participation of our seminar. So there are many participants from Germany, the blue sector on the left-hand side, from United States, from Italy, Spain, and United Kingdom. We were very happy for the interest in a wide audience of colleagues. And Dresden you find here in this yellow part of the world map. It's in the former eastern part of Germany, and it's actually at the same, located at the same river as our today's speaker, namely the river Elbe. And in the background of my picture, you see the Church of the Lady, which is one of the churches which has been rebuilt after reunification. We started our seminar last week with a talk by Thomas Hillen from Canada. And today, it's a great pleasure to welcome Klaus Pantel from Hamburg. Before he presents his talk, I would like to shortly introduce Professor Pantel, who has a background in human medicine from the University of Cologne. Interestingly, he did a PhD thesis in mathematical modeling, namely mathematical modeling in hematopoiesis, with a degree in Dr. Med. So he's a medical doctor with a mathematical thesis. He continued his habilitation, that's a German degree, kind of second PhD thesis from the Ludwig Maximilians University in München, in an area of immunology, immune biology, and aspects related to cancer. In his postgraduate career, he visited several places in Germany and the United States. And since 2016, he is a visiting professor at the University of Bergen, Norway. Today, Klaus Pantel is the director of the Institute of Tumor Biology, a full professor of medicine at the University Hospital Eppendorf in Hamburg. He received several prestigious awards, including twice a European Research Council grant, ERC Advanced Investigator Grant, and also in 2010, the German Cancer Award. He's publishing and has been publishing in many prestigious high-ranked journals and has today more than 550 publications. His research interests are multifold, centered around cancer, and I would say lately, particularly around circulating tumor cells. His talk today is on liquid biopsy, tracing of tumor cell dissemination in cancer patients at the single cell level. We are very much looking forward to your presentation. Before you start, I would like to make some technical remarks. The participants are invited to make chat questions in YouTube and use the Q&A option in Zoom. You are recommended to use Zoom. The technical quality we saw last time is better in Zoom than if you follow the YouTube channel. And after the talk of Klaus Pantel, we will collect the chat questions and possibly also today allow live questions. We will see how we deal with time. And now we are very much looking forward to your presentation, and we switch screens to your talk. Thank you for coming and talking in our seminar.
Well, it's really my pleasure. Thank you very much for this very kind introduction. And I hope that everybody is seeing my screen, which is the most promising challenge today, but I guess it worked very well. I can see it. And it's really my pleasure to talk today and give you an introduction on liquid biopsy and tracing of tumor cell dissemination in cancer patients. And I wish I could be at the beautiful city of Dresden today, but this is probably just the second best way of giving my talk. Well, liquid biopsy is actually there with a promise to give a comprehensive assessment of circulating blood biomarkers. And on this slide, you see how complex the situation is. In cancer patients, you have a primary tumor, which appears first at cancer diagnosis. And this tumor can shed cells and cellular products into the blood circulation. But these disseminating tumor cells can also end up in the lymph nodes and build up a lymph node metastasis. And from this lymph node metastasis, also cells can go into the blood. And in addition, the cells can travel to distant organs, such as the brain, the liver, or the bones, and form distant metastases in these organs. And these distant metastases can also set off cells and cellular products into the blood. So depending on the stage of disease, the cells in the blood can be derived from the primary tumor, from the lymph nodes, or from distant metastatic sites. In the blood, we actually trace cells, and we call them circulating tumor cells. They can be in the blood as single cells, but also as cell clusters. And we can also trace cellular products, in particularly nucleic acids or extracellular vesicles. So the concept of liquid biopsy is here. The definition is that we can analyze tumor cells, call them circulating tumor cells, or their products, in particular DNA, but also circulating microRNAs or extracellular vesicles. And the blood is our key tissue, but these technologies can be also applied to other body fluids. What is the rationale? The rationale is that in cancer diagnostics, usually tissue biopsy is the gold standard, but tissue biopsy, and particularly to biopsy metastasis, is sometimes very difficult because sometimes the tumor tissue is located at some remote places, such as the lung and the brain, and it's difficult to biopsy and put a needle into it. Even if you can biopsy a tissue, it's very well known that the different metastatic sites in one patient can have different genomic information. So there is considerable inter-patient heterogeneity, and just getting one single biopsy may not give you the whole picture. And the third argument is that you want to get real-time information, what happens in the cancer patient, for example, during natural progression or during therapy. So you want to get sequential information over time, and it's not very feasible to put a needle in a patient every week to find out what's going on with the tumor cells, but you can take a blood sample every day or every week and can monitor cancer patients. So the vision is that you get a comprehensive and real-time information by the analysis of blood or other body fluids. The next picture shows you the technologies that are used for the two most prominent analytes of liquid biopsy. These are circulating DNA and circulating tumor cells, and I will focus a bit more on the tumor cells today. But just to tell you that the DNA can be actually assayed from the fluidic part of the blood plasma, and there are, of course, tumor-specific characteristics on the ctDNA that can be used to definitely say that you have detected tumor DNA. The tumor cells, the CTCs, they are actually captured from the cellular part of the blood, and you have actually three different analytical steps. First, you need to do a CTC enrichment because the circulating tumor cells are in blood, one cell in the background of millions of blood cells, so you need to enrich it in order to see it. For the enrichment, you can either use biological properties of the tumor cells. For example, the tumor cells can express certain surface antigens, and you can pull out those with a positive selection mode. Or you can pull out all the leukocytes, we call it negative selection, and enrich the tumor cells thereby. You can also use physical properties of the tumor cells to enrich them. For example, the tumor cells can be bigger than the leukocytes, or they can be less deformable than the leukocytes, and they may also have some particular electrical charges. After the enrichment, you have the tumor cells there at a frequency 1 in a few thousand leukocytes, and you still need to do a single cell identification. And this identification you can do with immunocytological technologies. You can use certain tissue-specific markers, for example, keratins, to say this is most likely a tumor cell and not a blood cell. Or you can also use molecular technologies like quantitative PCR, detect tumor-specific transcripts. And in the third layer, you can also use functional assays, such as the EpiSpot assay, to show that the tumor cells secrete a tumor-associated protein. In the third, I think, layer of analysis, which is the downstream analysis of these CTCs, you can look what kind of characteristics do the cells in the blood have? Because not all cells in the blood are equal. There are good ones and bad ones, probably more or less aggressive ones. In order to further characterize them, you can do that at the genome, at the proteome, transcriptome, and functional level. At the genome level, you can look for genomic aberrations with different technologies. At the proteome level, you look at the protein pattern. At the transcriptome level, you look at the RNA pattern. And most likely, we are also now able to look at functional properties of CTCs. And I will give you a few examples of that during my talk. What do we do with that? Well, what we actually can do is we can track tumor burden in cancer patients. And this is a theoretical curve here, which shows you what happens from the start of the tumor at the time 0 to the development of distant metastasis. And you can see initially the tumor probably starts from one cell and grows out until it gets detected. Usually you have about 10 to the 9 tumor cells when it can be detected. And then, of course, the question is, does the local therapy, for example, the surgery, can cure the patient? In some patients, that is possible. You can get rid of the tumor cells by the surgery. But in some other patients, tumor cells have been already disseminated, and these cells can grow back. And they can come back, and they can cause something that we can call minimal residual disease, which can be detected with liquid biopsy methods. And this disease can then be probably treated again, and the cure can be tried again, or the disease can recur again and cause overt metastatic relapse. And this relapse then, of course, deserves more aggressive therapy, and probably there is really no cure at that time point anymore. So we can use now the tracking of the blood samples to get information of these different stages of tumor progression. Interestingly, we of course probe now the blood because it's easily accessible, but in our earlier work, we have looked at the bone marrow, which of course is also a very interesting organ. and a very interesting reservoir for disseminated tumor cells where they can hide from chemotherapy and other things, and they can also probably circulate from the bone marrow into other tissues. The bone marrow can be analyzed by taking bone marrow aspirates from the iliac crest of cancer patients and using keratin staining to distinguish the cancer cells from the bone marrow cells. We were able to find in more than 30% of breast cancer patients that were free of metastasis already disseminated tumor cells in the bone marrow, and more than 40% of patients with prostate cancer. Interestingly, I think that the DTC detection was correlated to metastatic and locoregional relapse. Most of the DTCs were non-proliferating, but they had a stem cell phenotype. And other groups found out that the DTCs can occupy their metastatic stem cell niches in the bone marrow, and that may give them a certain advantage to survive. Well, in patients like breast and prostate cancer, you expect cells in the bone marrow because bone metastases are frequent. But we also found in many other tumor types where bone metastases are not so frequent that you already have disseminated tumor cells there. So it seems to be that the bone marrow may be an interesting reservoir for disseminated tumor cells, even if the cells do not have the right conditions to grow out and form a real metastasis in that organ. Well, what else do we know? What we have learned over time is really that the interaction with the tumor cells and the bone marrow cells, for example, the osteoclasts, is very important to induce the growth of these disseminated tumor cells. We have also learned that we can get information on potential therapeutic targets to intervene with this crosstalk. We have also learned that if the disseminated tumor cells arrive in the bone marrow, and that's also true for other organs, the tumor cells that grow, for example, in the breast for 10 years and then end up in another organ, such as the bone marrow, they need to adapt to the new conditions in the bone marrow. This adaptation is important for their survival. We have also identified genes that were related to bone marrow dissemination. And one of these genes is the RI2, retinol acid-induced gene, that if it is upregulated, seems to act as a metastasis suppressor. So the dissemination of cells into the bone marrow seemed to be not just a random process, but seemed to be regulated by genes. We also found that in order to survive in the distant organ, the cells really need to induce or activate the unfolded protein response. It's a stress response. And I think the tumor cells that have the best stress response are probably the ones that can survive in this new organ. And last but not least, David Leiden and his group could also show very nicely that the primary tumor is not just releasing cells into the circulation, but already releasing extracellular vesicles, exosomes, that then go to distant organs and make specific niches that help the tumor cells to home in this organ. Coming back to our identification, we were asking ourselves, is it just a random process that tumor cells can release cells in the circulation, or is it a process that is regulated by certain genes? So we compared actually breast cancer patients without and with tumor cells in the bone marrow and did a transcription profiling. And out of this work, we discovered a new gene, the RI2 gene, that seems to be upregulated in patients which had no bone marrow involvement or downregulated in bone marrow positive patients. So it seemed to have a DTC suppressing influence. And to make a long story short, and that has been published, we found out that this RI2 can interact with another partner, the CTBP2, and this interaction can lead to some epithelial mesenchymal transition. And we all know that this EMT is a very important process in metastasis development. And this is shown here, we know today that the cells that grow as an epithelial tumor, for example, breast carcinoma, prostate carcinoma, lung carcinoma, colon carcinomas, they're all epithelial tumors, but they can change their phenotype in a more mesenchymal way, and that's called epithelial mesenchymal transition. And this transition helps them to disseminate. But we also know in order to form a solid metastasis, there need to be probably a reversal of this process, an MET. And this reversal makes a mesenchymal phenotype again into an epithelial phenotype, which allows then to build up a secondary tumor or a metastasis. So the cells which have the highest plasticity to go in one or the other direction are probably the most aggressive disseminated tumor cells. Switching now to the blood, because this is, of course, clinically the most easily accessible organ, much easier than the bone marrow. We actually performed a variety of studies in different tumor types, many studies in breast cancer, but also in other cancer types, which are shown here on that slide. And all of these studies have one thing in common, and that is actually that the CTC counts at initial cancer diagnosis were associated with unfavorable prognosis. And that actually means that the ability of a tumor to set up cells into the circulation, to actually release cells in the circulation, is a sign of a more aggressive tumor. So in clinical terms, we can use the CTC counts to enrich actually patients which have a more aggressive study, a more aggressive tumor, and that may actually help to improve the current cancer staging. And there is also already a designation, it's called CM0, which means there are no clinical signs of any distant metastasis, but I+ indicates that there are individual tumor cells present that can be detected with sensitive method and that pose a risk for later relapse. Let me just show you one or two examples of these studies. This study was done in breast cancer patients. And these breast cancer patients, they receive neoadjuvant therapy, which means they get some chemotherapy before the tumor has been taken out by surgery. And what we could actually find out is that the CTC count provided prognostic information independent from the response of the primary tumor. And that actually tells you that the primary tumor response to chemotherapy is one thing, but the response of the cells that disseminate are another picture. And to put both together gives you probably a high information on whether the therapy has worked in cancer patients or not. And that was validated, as you can see here, on more than 2,000 patients in an international meta-analysis. So the circulating tumor cells, interestingly, reflect the potential of a tumor cell to disseminate through the bloodstream. And in this international study, you can see that the effect of this is quite remarkable. You can see in the top graph, in the Kaplan-Meier curves, that if you have 5 or more circulating tumor cells, your survival is much worse than if you have less tumor cells. And in the table below the Kaplan-Meier curves, you see that the risk ratio, the hazard ratio to develop metastasis later on during the course of the disease was about 6 times higher if you have 5 or more CTCs. So quite a substantial effect. The other application is, of course, can we, after primary diagnosis, track the disease and can we detect residual disease in cancer patients? So can we perform a post-surgical surveillance by taking blood samples over time? And in this study, we have done a blood sample. We have performed the blood sample 2 years after adjuvant chemotherapy. And we could detect circulating tumor cells after 2 years. And these cells are not circulating from the primary tumor taken out by surgery, because the CTCs only circulate for a few hours. These cells are from some residual tumor cells that are not seen with other methods. And if you have such tumor cells in your body, you can see that you have a worse overall and disease-free survival. So these cells matter. So the prime question now is not only can we detect a signal for minimal residual disease, but can we also use that to improve our therapy? And together with Dan Hayes, we have suggested a need for so-called post-adjuvant clinical trials that can then be performed in patients years after initial therapy when they still have signs of residual tumor cells. Another application that is very important is, can we analyze the circulating tumor cells and say where they're coming from? Because it might be quite important for us to know, are these cells coming from the liver, from the lung, from the bone, or from the brain? A first attempt was done by an exome sequencing of single CTCs isolated from the blood of breast cancer patients with brain metastasis. And we are wondering, can we see some genomic signature that is in common? And that was actually the case, as you can see here on the profile shown on that slide. We had quite a few aberrations that were either gains or losses that were present in most of our CTCs. So there was quite some high clonality of the CTCs, and maybe we can see in the future which of these aberrations tell us that the cells are derived from brain. Brain is also in the focus of our interest, and the group of Hajo Wigman and her colleagues looking very carefully what are the biological factors that cause and contribute to brain metastasis formation. And that paper just came out. It shows very nicely that LCAM is involved in that process, and it actually acts through the interaction with the vascular endothelium. And if you're more interested in that, I can refer you to that paper. And what you see here very nicely in the picture panel below is that the expression of the circulating tumor cells shown in the upper panel was similar to the expression of the brain metastasis, again giving some information by a blood sample what's going on with the brain metastasis. The other, of course, application is really tracing tumor evolution from primary tumor to metastatic disease. And this is one example in patients with colorectal cancer. What we have done here is we, together with Katrina Lix-Panherbier and her group from Montpellier, we could get blood samples from the tumor draining vein, the mesenteric vein, and we could blood samples from the peripheral veins. And we could get access to the liver metastasis. Liver is the primary organ for metastasis in colon cancer patients. And then we were actually asking the question, can we see some genomic progression if we go from the mesenteric blood to the liver to the peripheral blood to the distant organs? And that was a work very nicely done by Simon Josse in our institute. And we could actually show very nicely that with each of these steps in the metastatic cascade, we had an acquisition of new genomic changes. The second study I would like to share with you was done in breast cancer. This is a patient with estrogen receptor positive breast cancer, quite a typical patient who got diagnosis around 1999 and got a metastasis about 10 years later. And these late recurrences are typical for estrogen receptor positive breast cancer. So we actually got the primary tissue. We got blood at the time of metastasis around 10 years later. We actually analyzed individual tissue fragments from the primary tissue, and we analyzed individual CTCs, both by NGS methods. And what we found is really summarized here. We could see that there are different areas in the primary tumor. So there's intra-patient heterogeneity in terms of copy number aberrations. But we could also see, comparing that to the CTCs taken at the time of metastasis, that some of these areas were not reflected at the time of metastasis in any of the CTCs, but some areas were very nicely found also at the time of metastasis in the CTCs. And these probably aberrations are probably more linked to the development of metastasis than the other. It also shows you if you blindly put a needle in one area of this primary tumor, you may get one or the other type of genomic aberrations. So the tissue biopsy can also lead you into wrong direction. A further application of liquid biopsy is, of course, the question, can we use it not only to make beautiful scientific studies and prognostic estimations, but can we also use it to improve cancer therapy? And for that, what we can do today is we can measure markers of therapeutic targets or resistant mechanisms, and we can do that on the circulating tumor cells and on the circulating DNA. And that allows us to look on one hand at genomic evolution, if we look at the DNA changes, or we can also look at transcriptional plasticity, if we look at transcriptional changes. And I will give you a few examples. For example, in prostate cancer, it's very well known that the androgen receptor pathway is certainly one of the key targets for therapy. And what has been actually known, what has been known here is that some of the agents that are used to target this pathway do not work when you have a variant expressed of the androgen receptor. And this androgen receptor variant 7 actually lacks the ligand binding domain and actually signals permanently. So what we have done is actually asking how heterogeneous is the expression of this resistant variant of the androgen receptor. And for that, we have done a multiplex mRNA profiling of single CTCs from this prostate cancer patient. And you can see that you can find different clusters of CTCs. And interestingly, if you look here at the heat map, each line is an individual CTC, each column is an individual transcript. And if you look for the ARV7 expression, it's really here highlighted. You see that you have some of the cells that have expressed the variant and some of the cells that have not expressed the variant. So there is resistance in terms of ARV7 expression. There's heterogeneity. And you can imagine that the cells that have the resistant receptor expressed will grow out under therapy. And this can be also made visible by another technology where we use in situ hybridization to look at individual transcripts in individual cells. And here we could actually see that in most of the cells, we could detect CTCs in these advanced prostate cancer patients. And quite a few of these samples, almost 80%, had actually this expression of the resistant variant on the CTCs. So indicating that there is certainly heterogeneity in expression, but it seems to be not a rare event. Another example for a therapy target that can be analyzed on the CTCs is, of course, the PD-L1 expression. We know that PD-L1 is actually expressed on the tumor cells. It interacts with the PD-1 ligand on the leukocytes and the T cells. And this interaction can block the immune system. And today, antibodies can be used to block these blockades. And the Nobel Prize was actually given for that discovery 2 years ago. So we were asking, can we detect actually these immune checkpoint blocker molecule on the CTCs? And the answer is yes, we can do that. And maybe this information can help in the future to say which patients will respond to the PD-L1 blocking therapy. The second, newest, question that we have is a bit provocative. And all of you who have ever looked at ERC grants, they know that the ERC always asks you to submit provocative grants. And they are one of the agencies that really mean that. So I wrote my second ERC Advanced Investigator Grant on the question whether the mobilization of circulating tumor cells occurs during cancer therapy. And I was not just referring to systemic therapy, I was just asking, or we are just asking right now the questions, does diagnostic biopsy, surgery, or radiotherapy, so the primary therapy, can lead also to a mobilization of tumor cells into the blood? Very provocative question. Do we have any evidence for that at all? Well, we have actually published a pilot study, which is shown here. Here we looked actually for CTCs in men that were undergoing prostate biopsies. You may know that if you undergo a prostate biopsy, it's not only one needle in your prostate, you usually get 12 needles into your prostate, because prostate cancer is a very heterogeneous disease, a multifocal disease. So you need to take many needle biopsies to be sure not to miss it. And we were wondering whether this procedure can lead to an increase in CTCs. And you see that in many patients, there were no change, it was 0. We had a few patients where we saw a small decrease in CTCs, but we had also quite some patients where we saw a very small or moderate increase in circulating tumor cells after the biopsy. And I think we were interested, does it really affect the recurrence rate? And this is a very, very early analysis. These are really pilot data on 74 prostate cancer patients with and without an increase in the number of circulating epithelial cells in the blood. But we see from this very early analysis that there seemed to be some correlation to outcome. And now we are really looking into much more depth into that to see whether we can confirm that with our ERC grant, and whether we can find signs that these cells are viable and contribute to tumor progression. The, I would say, most attractive application of liquid biopsy is probably the early detection of cancer. And of course, if you can replace the biopsy, the prostate biopsy or any other biopsy one day by a single blood analysis, that would be fantastic. If somebody would ask me whether I like to have 12 needles in my prostate or a blood sample taken, of course, the decision is easy. But we are not there yet. What is the challenge for early detection? Well, we really want to detect early tumors, so stage 1 or small tumors, not the big ones that can be detected with other means. The sensitivity is important. We want to have a high sensitivity and not miss too many false negatives. The key is, of course, also false positives. We do not want to tell a cancer patient that your blood test is positive, but there might be no cancer or no relevant cancer. And that specificity is a key thing, because imagine you screen the whole population of Germany and you, or another state, you would have a 99% specificity, which sounds fantastic, but 1% false positive. Then if you screen 10 million, you have 100,000 inhabitants that you call cancer patients, but that is wrong. And you can imagine therefore that you need very high specificity of these screening tests. Also, if you see publications, always look whether the healthy controls are age-matched and whether people have also analyzed confounding non-cancer diseases that may increase and give a false positive signal. So this is not just theory. A few years ago, colleagues published actually some interesting results. They could show that in elderly patients, and many of us belong already to that group, there is a background of cancer-associated mutations in non-cancer control patients, and they called that CHIP. And this clonal hematopoiesis of indeterminate, I think, prevalence is actually a problem, because it has been associated with a slight increase to develop some cancer disease, but most of these patients are not developing cancer. So you need to be careful that not every mutation you analyze in the blood could be really the signal of an unknown cancer. So lately, I think there are quite a few groups who now try to find the better blood test, and they also combine different analytes. And in this publication a couple of years ago by the Johns Hopkins group, they looked actually at circulating DNA from 16 genes and about thousands of mutations, and they combined that with 8 protein biomarkers and then a machine-based learning approach. And this effort was able to detect with high specificity and sensitivity some tumor types, such as ovarian cancer or colon cancer, but for breast cancer, it still had quite a low sensitivity. And also, if you look at early stages, the sensitivity was not so high. So they went on with that, and they are further improving that now. In particular, I like that approach because I believe that the combination of different liquid biopsy markers could actually increase the chance to have a good early detection blood test. So this kind of combinatorial approach is probably a good. And the liquid biopsy family is growing. We have circulating tumor cells, we have the cell-free nucleotypes, the extracellular vesicles, but we also have now the tumor-educated platelets introduced by Tom Würdinger's group. So these kind of combinations may allow us, together with artificial intelligence, to find the right compositive biomarker for early detection. And actually, we are very active in that field. We have a EU Marie Curie Slomska network called ELBA, which is coordinated by Tom Würdinger. And we have another network on early detection of prostate cancer in the ERANET transcript scheme of EU projects, which is coordinated by me. Interestingly, if you want to look at early detection markers, it's very important to have a good setting. And in Hamburg, we started a few years ago the Hamburg City Health Study, which is a cohort study. And here, 45,000 individuals between age 45 and 74 are actually coming to the hospitals. A lot of material is sampled, also blood, and a lot of data. And I think that this cohort study will be a wonderful asset for early cancer detection studies, but also for early detection of other diseases. So what can CTCs also do for you besides the characterization? You can also look what are the functional properties of the circulating cell. And here we are just at the beginning, but at least the functional age has started for CTC research. A few years ago, we could actually show that we can take blood from breast cancer patients and inject the cells into immunodeficient mice and form tumors. These were the first xenografts. A little bit later, we were also able, and particularly the group of Katrina Lix-Panabier, to induce cell lines, establish cell lines from CTCs, and these can help us now as models actually to understand CTC biology. So just lately, we have developed a new cell line from an ER-positive breast cancer patient, and this cell line is also able to grow after intramammaliar injection into immunodeficient mice, and we can also get metastases at organs that are relevant in breast cancer. So this is a wonderful new tool to understand the biology of CTCs in breast cancer. However, we should also keep in mind, if we do xenografts, we inject them in immunodeficient mice. So in order to learn something about the immune system against CTCs, we probably have to develop immunized models. So in conclusion, I think that the CTCs and ctDNA provide complementary information for liquid biopsies. I think the assays always need to be thoroughly validated by independent expert groups. And I think what we need to know now is how can we use this approach to change the therapy of patients, and we call that interventional clinical study that are actually based on the liquid biopsy result. And then we have to demonstrate that any kind of change of therapy based on the liquid biopsy result is increasing the lifetime of patients. So how can we get there? How can we go from a beautiful publication or a beautiful list of publication to get a biomarker into clinical routine? And this is really a very, very stony road. It's not that the Nature paper by itself will create a product for the cancer patients. So the EU gave us actually 5 years of support to validate existing CTC biomarkers and ctDNA biomarkers and others. So it was focused on liquid biopsy, and it was an interaction between academic groups and industry. So we focus actually on blood-based diagnostics in lung and breast cancer, validation of existing assay, and we were 40 partners throughout the EU. The program has been finished in December last year, and we had established this fantastic network. And in order to sustain it beyond the funding from the EU, we established our own network called the European Liquid Biopsy Society. And this is now open also for new members. So if you are interested in liquid biopsy, let me know. We are happy for new members that join our liquid biopsy consortium. I think the goals are really on that slide. I don't want to go through it in all detail, but I guess the key aim here is to foster the introduction of liquid biopsy to clinical practice, encourage interactions between academia and industry, provide actually also training and dissemination of good work and also standard protocols, and make sure that we really all together can bring this interesting biomarker concept into something that helps at the end the cancer patients and not just our CVs. So this is just one slide I would like to show. What are the efforts in the different areas of the world on biomedical research? You see that there's an increase in biomedical research spending in Europe, in the United States, also in Germany. But you see there's a very exponential increase by China. We don't know how that will be now after COVID, but I think that it's very interesting that we need to invest into biomedical research today. I also think there was a very interesting issue of Nature. If you're interested in science in Europe, you can look at the Nature volume from May 23rd last year. There are a lot of interesting charts on science in Europe, which I think are very informative. And there's also a list of the top 200 healthcare institutions in biomedical science. And of course, we at the UKE were very happy to see us in that list, which I think was fantastic for us to know. And but I think the major issue here is, if you're interested in research in Europe, these charts that are really published in this issue are very informative. So at the end, I would like to say that research is, of course, teamwork. It's not just the work of one person or a small group. We have developed a liquid biopsy research network at UKE between institutes that work more on the basics and clinics which are doing the clinical studies. This translational interaction between basic and translational and clinical research is fundamentally important for the success of this research program. And at the end, I would like to show a picture of our team at the Institute of Tumor Biology. We are around 50 researchers from 14 different countries, a bit like United Nations. And I'm very happy to have actually the opportunity to work with this fantastic team. We are also supported by national and international granting agencies, and I'm very, very happy to show that. And I really would like to thank these agencies a lot for supporting our research over the past years and decades. Thank you very much.
Hello?
Yeah.
You understand me?
Yes.
Okay, thanks a lot for your inspiring talk. And we have now time for questions. And we want to test if the live question is possible. So may I ask Thomas Hillen to put his question. Can you unmute your microphone, Thomas, and ask your question personally? Okay, it doesn't work immediately, so I will ask the question of Thomas Hillen to Klaus Pantel. What do you know about the dynamics of CTC in the blood? Can they be attacked by NK cells? Could they be protected by platelets or blood clots?
Yes, actually, the interaction of CTCs with other cells in the blood is probably under-investigated, but incredibly important. We know that the blood platelets can cover circulating tumor cells. They can protect them and also may help them to extravasate. We know from the work from Nicola Aceto published last year that the CTCs can interact with neutrophils, and these neutrophils can change the expression pattern of the CTCs and may help these CTCs also to survive. So these clusters of neutrophils and CTCs seem to be biologically very important. We also, of course, assume that the CTCs can be attacked actually by immune cells. We have just recently published a paper showing that the PD-L1 expression is changing when the cells are subjected to higher oxygen levels in the blood. So maybe the blood passage is a bit like the Achilles heel of tumor cell dissemination. And the interaction with the cells in the blood are incredibly important. I have no doubts about that.
Mm-hmm. Thank you. We have a question from Simon Züger, Dresden, Berlin. What is the biggest challenge in using CTCs to improve the clinical outcomes of patients, and why?
Well, the biggest challenge, I would say, is to combine the right liquid biopsy assay with the right therapeutics. In order to change really the outcome of patients or the prognosis, you need to show that your measurement in the blood will change your therapy, and that this change is really leading to an improved clinical outcome. So there are two major variables. One variable is how good is your diagnostic test, and the second variable is how good is this change in therapy. So we have, for example, one very negative example. There was a study when the CTC count was used to decide whether breast cancer patients get another type of chemotherapy. But this study had a problem because when the CTC count went up, it showed that the patients were resistant to the first chemotherapy, but these were probably the most aggressive patients, and they were also resistant to the second chemotherapy. So at the end, it was blamed to the CTC test because people said the test was not able to decide whether they need another chemotherapy. But probably the truth is that the other therapy, the alternative therapy, was not really an alternative. It was just another therapy that didn't work. So the biggest challenge is to combine the right diagnostic test in a clinical setting that gives you a chance that the change in therapy is also improving the patient's outcome. So there's a strong link between diagnostics and therapeutics if you want to be successful.
Thank you. We have a question from Mexico, from Josue Nava Cedeño. What is the probability of developing metastases when CTCs are already detected in the bone marrow? Can you calculate or give the risk?
Actually, we have, for example, in breast cancer, seen that there is a significant increase in risk. I don't know the hazard ratio out of my head, but that's published in Brown et al. In the England Journal of Medicine 2005. It's one of the largest studies. But we have also seen that quite a significant fraction of these patients with DTCs in bone marrow do not develop a relapse within 10 years after a follow-up, right? So we can say it poses an additional risk to develop metastasis, but it's not a sufficient, let's say, condition, okay? So in quite a few patients, the body can control these cells. And we have to learn how the body is able to control these cells. How can they induce dormancy? Because this is really a fascinating field of research where we could get new information on very, very different ways of therapy, maybe.
Mm-hmm. Thank you. Another question. as liquid biopsies from primary tumors can be considered as the intermediate step prior metastatic dissemination, could one expect a much higher tumor burden in CTC samples versus primary tissue biopsies of the same tumor patient?
Yeah, that is possible. Absolutely. I think there can be definitely differences. We also, I think, one of the total unknown in this calculation is there high or is there any incidence of recirculation? We can measure the primary tumor quite well, the size and also the biology. We can measure the number of CTCs in the blood. We can extrapolate that to the total blood. We can also measure changes of that in the blood. But we don't know if a CTC disappears from the blood. Is it because the cell is dying? Or is it because the cell is extravasating, which is biologically something very different? And is there also re-extravasation or intravasation or recirculation, you know? Is it just like the cells go into an organ and then they go out of the organ? For example, hematopoietic stem cells in bone marrow are doing that. There is a pool of circulating stem cells and they always go in and out in a certain way, you know? So there are really some unknowns in this variable, but here I would say maybe some modeling could be quite interesting to see what kind of degrees could there be of these unknown processes and how can we really measure them better.
Thank you. We have a question from Luxembourg, from Simon Niclou. Do you see generally a correlation between CTCs and ctDNA? Has this been checked?
There are some papers that have looked at that, and there is, only a weak correlation. I mean, it depends, however, very much on the tumor type and on the disease stage. So, for example, if you go in breast cancer with bone metastasis, you can have a nice number of circulating tumor cells and also good levels of ctDNA. But we always have exceptions of that rule where we see a lot of DNA but no CTCs, and the other way around, a lot of CTCs but low levels of ctDNA. So I would not say to date that there is a tight correlation between CTCs and ctDNA, but we definitely need more studies where both parameters are measured with up-to-date technologies.
Thank you. We have a question from Pakistan, from Azhar Usmani. It was a very interesting and comprehensive talk, so it's a compliment. Even not being a biologist myself, I understood almost 50% of the technical problems and the potential of the proposed solutions. My question now is, how can I participate as a computer scientist at any stage of the proposed solutions for the CTC problem? Where do you expect a computational scientist to work on, particularly?
Well, I definitely think that it would be very good to model some of the changes, you know, that happen during tumor progression. We still know very little about the actual rate of tumor cell release per day or per week or per month. We know not much about actually the success rates of cells that extravasates, how many of them make a metastasis. I mean, we always have little snapshot information from our samples, but I guess to get some more dynamic insights, what is actually happening, that would be definitely something where, I mean, I could at least envisage that you could help, yeah. And thank you for the compliment for the talk.
So you wrote your PhD on mathematical modeling of hematopoiesis, but then after all the references you showed us today, there was not much mathematics. So
yes, I abandoned mathematics. Actually, I went from the mathematical modeling into experimental hematology, so in experimental work. And from that, I went into the cancer field. And unfortunately, I left that field. And fortunately, after many decades, I meet again people who are interested in mathematics. That's good.
Okay, thank you. So there's another question.
But you have to also understand, when I finished my thesis in 1987, whenever you gave a talk about mathematics, and bioinformatics didn't exist at that time, probably. I mean, you were really a stranger, I would say. And it was not easy at that time. I think today, I mean, bioinformatics, mathematical modeling, and systemic medicine, system medicine, all these are very, very interesting and hot fields. But in 1987, it looked really strange, I can tell you. So it was not easy to go on with that in my career, but I'm extremely happy that I now cross the road of mathematicians. That's fantastic.
Okay, thank you. We have another question to you. Could sustained blood biopsy promote the progression of tumor?
Oh, can you say that again?
Could sustained blood biopsy promote the progression of the tumor? And I think you answered this to some extent, no?
Yeah, I think that we don't know now. We don't know now. I mean, we have started some work and we will have now 4.5 years time with the grant, you know, to look into that. We all feel that we need some biopsies. There's no question about that. And but on the other hand, we know very little whether it really increases the release and whether the cells that are released are relevant or whether they are not relevant. But this is exactly what we like to find out the next 4 to 5 years. Yeah.
So you started out your talk with this hallmark of cancer heterogeneity. If you do now liquid biopsies of different liquids, what is heterogeneity in the different liquids?
Yeah, I
blood,
think we have,
lymph.
Yeah, I think that people, I mean, in the blood, we also have heterogeneity. We have not, to be honest, it's a very good question. We have, I mean, I don't know anybody who has very roughly analyzed the heterogeneity in the blood compared to the primary tumor. But we are probably now able to do that in the future because we can, for example, do diagnostic leukapheresis to get a sufficient amount of tumor cells, because you really need quite a few tumor cells. And then we may be able to see what is really the heterogeneity index of the CTCs compared to the primary tumor. We also have sometimes patients which have hundreds of tumor cells in the circulation, but these patients are usually advanced cancer patients, and then it might be not so easy to compare it to the primary disease. But I think we see, for example, that for certain markers like the PD-L1 or the HER2, we see some kind of signs of selection, I would call it very carefully, you know, in the blood.
Thanks a lot. I would like, Simon, can you try your question to put it live? Does it work? Unmuting the microphone? No, again. So I asked the question of Simon Züger. How similar are the genetic profiles of CTCs compared to cells from the primary tumor?
Well, we have actually done this comparison, and we find similar genomic aberrations between both, but we also find distinct differences. And I showed this one patient where we microdissected different areas of the primary tumor, and depending on the area of the primary tumor, the overlap was low or it was high. And this is quite interesting because it goes back to the very old papers, you know, of Josh Fiedler and others, you know, who have always claimed that there is this metastatic subclone in the primary tumor. And that's probably true. We can see that. So whenever people publish, let's say, a bulk comparison of a bulk tumor with the bulk CTCs, they may miss something, okay? So there is probably some spatial information in the tumor that needs to be delineated if you compare that to the CTCs.
So if you would calculate a phylogenetic tree of liquid biopsy tumor and the solid tumor, you would get similar tree or very different?
We would get for some areas of the primary tumor, we would get similar trees, and for other areas, we would get quite distinct trees. Yeah.
Okay.
It's very interesting. I think definitely there is development over time, because sometimes, as I showed on one slide, there can be 10 years between the primary tumor resection and the metastasis, you know, so there is therapy in between. So I think it is probably very likely that there is some kind of selection going on in this 10 years.
Okay, okay. Yes, I think we had a very nice discussion and talk, and I would like to thank you again for your talk, for the inspiration. The discussion has started. It can be continued in the Slack channel, for example. And before we close the seminar, I would like to give an outlook for next week. I would like before to applause to Klaus Pantel,
well, thank you very much.
Representative for the audience in Zoom and YouTube. And now I show you the. Can you see my screen?
Yes.
Good. Okay. So next week we will have Rob Noble, and he speaks on characterizing and forecasting of tumor evolution. So we will ask Rob if he can use the data of Klaus Pantel to improve his evolution broadcast. Thanks for joining the seminar. Distribute, if you like the seminar, this information about the seminar to your colleagues. And I hope to see you all again next week, next Wednesday, for Rob Noble's talk. Bye-bye.
Super. Thank you very much.
You're welcome. Bye-bye.